Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Organ on a Chip, Improving Preclinical Studies for Faster Drug Development. My name is Edward and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future streaming. A big thanks to CN Bio who helped develop the content for today's webinar. CN Bio Innovations develops devices that boost the precision and speed of biological research. By predicting the effects chemical and biological substances will have on human organs, researchers can fast-track improvements in healthcare. In the past 4 years, CN Bio has worked on more than 25 projects with pharmaceutical partners using the company's organs on chips and related microfluidic devices to gather precise human-relevant data. CN Bio is also collaborating with the U.S. Food and Drug Administration's Center for Drug Evaluation and Research. FDA and CN Bio are using CN Bio's organs on chips as a platform for drug development and regulatory evaluation, including safety testing prior to new drugs entering clinical trials. And now I'd like to introduce our speakers for today's webinar. Dr. Emma Skeets is CEO of CN Bio Innovations, which produces organs on a chip devices for drug discovery and research. She has steered CN Bio's commercial negotiations in more than 25 successful pharmaceutical company deals and overseen academic research and licensing programs that have led to the development of the company's leading technologies and IP. Dr. Skeets was responsible for licensing several key organ-on-a-chip patents from the Massachusetts Institute of Technology and other leading institutions, including Vanderbilt and the University of Oxford. Her team has developed the MIT technology into a commercial organ-on-a-chip platform that flexibly enables both single-organ and multi-organ experiments. Dr. Skeets was also a presidential scholar at MIT and holds a doctorate in chemistry from Oxford. Dr. David Hughes joined CN Bio Innovations in 2010 as head of engineering and was promoted to chief technical officer in July 2014. He oversees the company's research and product development programs, including major collaborative initiatives with academic and industrial partners. Dr. Hughes was a principal investigator on a $26 million U.S. Defense Advanced Research Projects Agency contract led by MIT, under which the team developed a 10-organ human body on a chip. He previously led a €4 million EU FP7 program which developed stem cell-derived models of metabolic liver disease. Before joining CN Bio, he worked in manufacturing and research and development at GlaxoSmithKline. Dr. Hughes graduated from the University of Oxford with a Master's in Engineering of Science and doctorate in Chemical Engineering. And now, without further ado, it is my pleasure to hand the presentation over to Dr. Emma Skeets.
Edward, thank you so much for that kind introduction. And thank you for all of you for joining our webinar today. It really is great to have so many of you join us for this afternoon's presentation. And I think it's a really telling sign of the growing interest from industry and academic labs in organ-on-chip technologies. In the last 10 years, I'm sure I don't need to tell this audience that there has been a real explosion in the publications in the organ-on-a-chip field. And this means that there has really been a growing evidence base to support the use of microphysiological systems in diverse applications in pharma, in basic research, in the evaluation of chemicals, cosmetics, tobacco, and other agents. Here's some of the publications that CM Bio has been involved in the publication of. And of course, there are many more examples that you can find on the web, on CM Bio's own website, or by following us on LinkedIn. So organs on chips have been hailed as this promised land of more translational biology and less animal testing. And there's been much less talk of how research labs outside of the major organs on chips centers of excellence can get access to and get the benefit of these novel technologies. There's also been much less talk of how pharma and academic labs are going to deal with the practical issues of validating these technologies, either to complement or potentially to replace research and workflows that are already implemented in their labs. And this really, I think, is where organ-on-chip vendors like CM Bio have an important role to play. We have to translate these innovations from academic labs into robust commercial products that are suitable for more routine use in research labs across the world. I view this as really our job, being to build the bridges between innovation and application. And the way that we're going to try and do that for you today is to take a very practical look at what it takes to implement an organ-on-chip system in your own labs. And by providing examples, some publications, and advice on how you can go about setting up and running successful organ-on-a-chip experiments in your own research. I'll say now that we're going to be using a couple of very short videos during the course of this presentation. The streaming quality of these videos will depend on the speed of your internet connection, but don't worry if the videos are a little bit stilted or they don't play especially well. They're only going to be less than 30 seconds each, and these videos and more are going to be available online immediately after the webinar, and we'll be providing a link to these videos and the materials that we share with you just immediately after this presentation. So before we dive into the main presentation, and much in line with Ed's comments for this webinar to be a very interactive event, we thought it would be interesting for you all to share your experiences of organs on chips by answering a very quick poll question. And at this point, I'm going to hand over to Ed, who has the technology in his hands to do this.
Thank you very much, Dr. Skeets, and that is correct. We do have our first poll question of the day. Our viewers can vote on this poll in real time, so we do encourage everyone to do so. Today's poll asks, what has been your primary method for learning about organs on chips? And the answer options for this poll are web search or social media, scientific literature, peer-to-peer learning, workshops, user groups, and/or demonstrations, or lastly, do you use your own research as your primary method for learning about organs on chips? And it looks like most of our audience members have voted, so I'll now like to close the poll and share the results here. We have 44% of the audience voting that scientific literature is their primary method for learning about organs on chips. A tie for peer-to-peer learning, 12% voting, and also another 12% voting web search or social media. 27% of the audience voting that they use their own research, and 5% of the audience voting workshops, user groups, and/or demonstrations as their primary method for learning about organs on chips. And with that, Dr. Skeets, I'll pass the mic back over to you.
Thanks for that, Edward. And I really appreciate everybody taking part in that mini poll. So we ran something similar at the outset of a workshop we held very recently in the last couple of weeks. And as expected, you know, the scientific literature continues to be a really important means for scientists to learn about these new techniques and devices emerging in organs on chips. I think that for more people to be adopting these types of technologies, the availability of kind of workshops, user groups, demonstrations, webinars like today are going to be pretty important. So hopefully the slides that we're going to share with you over the next 35 to 40 minutes are going to be instructive. And at this point, I'm going to hand over to CM Bio's CTO, David Hughes, to walk you through some slides.
Thank you very much, Emma, and thank you again to everybody for joining us on this webinar this afternoon. So firstly, let's start by having an understanding of what an organ on a chip, or indeed a microphysiological system, is. I prefer the term microphysiological system. I think it's more accurate, but you will also hear the term organ on chip widely used, and I may use them interchangeably during this presentation. So microphysiological system is a precision in vitro culture that aims to recapitulate aspects of human physiology and disease. That system will typically feature human cells or primary cells. It will usually incorporate some form of scaffolding or 3D culture. Very often there will be a fluid flow or microfluidics aspect, and indeed you will see that in some of the systems that CM Bio have produced. And lastly, the organ may require, or the organ mimic may require, some mechanical stimulation. And again, there are plenty of examples in the published literature of mechanically stimulated MPS or organ on chip systems. So to introduce our variant of organ on chip, the Physio Mimics T1 system, and this system we have developed and there's a confluence of experiences we've had in working with pharmaceutical companies, but also in undertaking the DARPA-funded Physio microphysiological systems program, which was led by Professor Griffith at MIT. So the system that we have built aims to be both simple and flexible. Our chips have an open well format, so in many ways are like a multi-well plate, but with the additional benefits that 3D culture and fluid flow can build. We've been very conscious about the materials of construction that we use in these platforms. We have also endeavored to make these platforms as easy to use. So during the cultures, we see minimal interaction between the user and the cultures. And we know as well that space in labs can be limited. So we've worked hard to minimize the footprint of the device and try and make it compatible with standard laboratory equipment. So here is an example of fluid flow being used in one of our systems. This particular example I've selected is our liver on a chip system. And in our liver on a chip system, we have an embedded micro pump which acts to drive fluid around a circuit. And in this particular implementation, the fluid flow is driven through a scaffold which contains the cells, and you can see that in the middle panel here. And on the right, you can see a schematic of how the tissue is arranged in 3D and the flow passes directly through that tissue. In other forms and to recapitulate other organs and tissues, the flow is moved in different areas and with different patterns around the cells. But hopefully this gives an example of how flow is used in these systems. So moving to the workflow for using the Physio Mimics system, and this workflow is very analogous to the workflow for many other in vitro assays you may well be familiar with. So the consumables for our device are provided sterile. These are placed into the device, and then cells are seeded and cell culture media is added. The cells are then transferred to the incubator for culture, Typically, with these cultures, and taking again an example from the liver, you will pre-culture the cells for a number of days to allow morphogenesis to occur before removing the cells from the incubator, taking them back to the microbiological safety cabinet, and dosing compound or other test articles. Then there will be a subsequent dosing period before the experiment concludes with analysis, and that may be again analysis of cells or analysis of cell culture medium. So I'll now show a video. Once again, hopefully this shows well for everybody, but if not, you can find it on the web. So here you'll see a device being removed from its sterile packet. The device is then loaded into one of the organ-on-chip devices using that simple handle mechanism. Like multi-well plates all over the world, the top is then removed and cell culture medium and cells are added. And this will vary slightly between organs, but the process is always pretty much as you see there. Okay, so moving on to the culture phase of the experiment. The organ chips are housed within a standard incubator on a docking platform. So you would move between the microbiological safety cabinet and the incubator and dock the device into the dock. We think it's very important and have worked very hard to consider the usability aspects of these devices. If things are difficult, fiddly to use, tubing, connectors, pumps, these kinds of things, that will not enable a robust and reproducible assay. So we feel that in the PhysioMimics device, we have incorporated many design features which enable usability, which I think consequently will enable robustness of experimental work. Next to the dosing and analysis phase of the experiment, and so here some points to pick up are the open well format obviously gives easy access to the cells, and that can be for dosing, sampling, and then to remove the cells at the end of the experiment for analysis. When we're dosing compounds, we're very sensitive to the materials of construction of these devices. We know that, for example, PDMS can lead to non-specific binding of small molecule drugs. So PDMS has been eliminated from our devices. And so then we shall move to an example of a more complicated system. So the architecture that has been developed is appropriate and can be used with either single organ systems. So, for example, the plates you saw on the previous slides were some of our liver on a chip plates, but the same architecture will also allow you to use multi-organ or interconnected organ systems using the same hardware. And very importantly, our philosophy has been to make these devices open source. So here we try to enable, or we do enable, the onboarding of models developed by others. And you will see examples later in the presentation of where that has occurred. So we don't seek a one-size-fits-all here. These platforms will allow the models that you have developed in your own research to be brought together with the platform and linked to other organ systems. So lastly, I'll show a short video relating to the installation of the technology and hopefully convince you that these technologies can be very easily and quickly brought into your laboratory and be brought up and running. So here you will see one of my colleagues installing the dock into a standard incubator. The controller, which sits outside the incubator, is linked by a single set of cables to the dock within the incubator. These are simply plugged in using push-fit connections, and then the device can be placed on and the system is ready to use.
Wonderful. David, thank you for that. I appreciate that is a rapid tour of just one example, our own PhysioMimics system. but hopefully it gives you a good sense of what it is to bring these technologies into your own laboratory. And as I say, these and more videos are available online on our website at cnbio.com. So before we move on to sharing some examples, publications, and guidance around running successful organ-on-chip experiments, we thought that your answers to a second quick poll question might help David to tailor some of what he'll say for today's audience's interests. So, Ed, I think you're going to run this poll again for us.
Absolutely. Thank you for sending that over to me, Dr. Skeets. We do have another poll here for our audience. Again, viewers can vote on this poll live in real time. This poll question asks, for which organs or organ systems are microphysiological systems most relevant to your research? And the options here are the lung, the gastrointestinal organs, for instance, gut, liver, pancreas, the target organs of toxicity, cardiac, liver, kidney, brain. Is it other, or the last option there, multi-organ as your microphysiological system that's most relevant to your research? Again, viewers can vote on this in real time. One more time, for which organs or organ systems are microphysiological systems most relevant to your research? Is it the lung, gastrointestinal organs, target organs of toxicity, other organs, or multi-organ? And it looks like most of our viewers have voted on this poll, so I'll now share the results with everyone. 33% of voting target organs of toxicity as the microphysiological system most relevant to their research, 29% voting gastrointestinal organs, 22% voting multi-organ, 11% voting other, and lastly 4% voting lung. Very interesting results there. I'll pass the mic back over to you for some comments, Dr. Skeets.
Ed, thank you for that. Great. Well, what I hope through the next few slides that David's going to share with you, we're going to give a couple of examples from our research in the modeling of the liver and liver disease, but also some multi-organ examples incorporating barrier models, considering factors like organ scaling, the use of modeling to interpret results, and so forth. So hopefully this is all going to be very broadly useful across the research interests that have been expressed there.
Thank you, Emma. So I will touch upon 4 examples, the first 2 being single organ examples, the last 2 being interconnected systems. So talk about building a healthy model of the liver, then look at producing a disease pathology again in the liver, look at a relatively simple multi-organ model that incorporates two organs, the lung and liver, and then finally touch a little upon the work done under the DARPA consortium led by Professor Griffith to build a 10-organ on a chip model, or a human body on a chip model. So I think it's important at the start, and when we consider building models and assays, to understand that the organ-on-chip system forms part of an ecosystem, and all of the components in the ecosystem need to be considered to produce a good model. So taking here an hepatic model as an example, but this is fairly generic. You know, we need the primary or parenchymal cell of the organ. We may need to incorporate non-parenchymal cells and perhaps even immune cells in the model. The cell culture medium is again of importance, and I'll show an example of where inappropriate choice of cell culture media components might affect results. And then, of course, if we choose to create a disease model, we need some disease-causing agent. So that might be fat, it might be a virus, and all of those things, I think, are well tied together by the organ chip, which sits at the center of the model. So first, the relatively more simple model. So here we're building a primary human hepatocyte model of essentially the healthy liver, and the couple of components that are interesting are the cells, obviously, and also we should think about the cell culture medium that we use, and of course the organ chip model that they're housed in. So here you will see an example of data generated in our lab and in the methodology we use to form our 3D tissue structures. So in the liver on a chip system housed within Physio Mimics, there is a scaffold. The cells are seeded as single cells into that scaffold, and over a period of around 3 days, they undergo a morphogenic process, moving from single cells to what we would term a micro tissue unit. And the scaffold has an array of these micro tissue units. And on the right-hand side, you can see some images that show polarization and bile canalicular formation of the hepatocytes within the micro tissue. And one of the aspects that is particularly important, especially when working with primary cells, is to understand the donor-to-donor variability that is inherent in working with primary cells. In our liver on a chip model, we've worked with over 50 lots of plateable grade primary human hepatocytes, and you can see some images here, and then on the right-hand side some cell health data looking at the differences between donors. And I think that this is an important point to stress, that when working with these systems, one must consider the donors available and the supply of donors that can be used in the system. So if we move on to an application of a healthy liver model, you'll see here a paper that we co-published with colleagues at MIT looking at cytochrome P450 metabolism of a number of compounds. You can see there 6 compounds across a number of different donors. And I think 2 interesting aspects to highlight about the study are that the organ chip device indeed maintains the difference in phenotype of the donors. So we do not get, each donor's phenotype is held within, we do not get a convergence of phenotype over time. And that could be important for studying metabolism, but might also be very important later if one wanted to study disease processes and perhaps used patient-specific material in these kinds of models. The second point of interest is that data from these models can be used to do in vitro, in vivo extrapolation or correlation, and you will see some data on the right-hand side here for lidocaine, showing a population-based model and demonstrating that the data generated in the liver on a chip can well replicate the in vivo pharmacokinetic profile seen. So to move on to the next model, which is our fatty liver disease and NASH model. So when building this model, additional components were required. So for a fatty liver disease model, inflammation and fibrosis are important. So non-parenchymal cells of the liver were included, primary human Kupffer cells and primary human stellate cells. Obviously, we need a disease-causing agent in this case. So we have modeled the disease using both free fatty acids, so fat, but also using inflammatory stimuli like lipopolysaccharide, LPS. So just some brief introduction on the pathology of NASH. This is, I think, a coming disease, particularly in the developed world, and moves from a starting point of relatively benign steatosis, which is accumulation of fat within liver cells, moving through to NASH, which is characterized by inflammation, and then latterly fibrosis, and then lastly moving on to cirrhosis and ultimately potentially liver failure. So in building the model, we were interested to try and recapitulate those stages of disease. So here's some images of the microtissues, and here we've labeled the Kupffer cells and stellate cells within the microtissues. So the bulk of the cells, you can see here, the dark mass are the hepatocytes, and then the Kupffer cells and stellate cells sit within that, that mass of tissue. Now, as I noted earlier, the choice of media composition is important in these kinds of experiments, and here is some published work with the Tannenbaum group at MIT, where we look at the effects of hydrocortisone on inflammatory response. Hydrocortisone is often used in hepatic cultures to assist in the maintenance of differentiated state. And we can see here that works very well for hepatocyte cultures, but if we use excessive amounts of hydrocortisone in co-cultures that feature Kupffer cells, the resident macrophage of the liver, we find that the presence of hydrocortisone blunts their inflammatory response to LPS. So this highlights, I think, the importance of thinking carefully about what the media components are that go into these systems, and that becomes even more relevant when we think about having multiple organ systems connected together. So moving on, here is some of the phenotypic and pathological data that we see from our liver on a chip fatty liver disease NASH model. We can see on the left-hand side that the cells take up fat, and you can see that this is an oil red stain. You can see that from the redness of the fat sample. We can see that the presence of fat in the model starts to modulate the inflammatory response, so you can see additional IL-6 production in the fatty model. And also, interesting and importantly, we start to see that if we incorporate stellate cells into the model, we see pro-fibrotic behavior, so that's alpha smooth muscle actin and collagen production. And here, again, an important aspect, of course, is to attempt to reproduce clinical or animal results using these chip devices. And here we can see some data for obeticoic acid, which is an FXR agonist, in clinical trials for NASH. And indeed, if we give obeticoic acid to the cultures, we see the expected upregulation and production of FGF19. We see CYP7A1 response, and we see the obeticoic acid having an anti-inflammatory effect, so that there's a reduction in IL-6 production, but we don't see any change in albumin production, indicating that the compound is not cytotoxic in the chip. And just wanted to highlight here some of the ongoing research we have in this area. So we've been funded by Innovate UK to take the NASH fibrosis model that we've developed and work in collaboration with AstraZeneca to use the model to undertake a systems biology approach. So we will build a detailed systems biology model of the liver on a chip NASH model, and then we will use that in target discovery activities and potentially in the hopes of repurposing compounds. So I think this is an interesting and important example of these kinds of technologies starting to have a real impact in the pharma development pipeline. So moving on now to some multi-organ examples, and these again are implemented using the same Physio mimic system which I showed you earlier, but this time a different consumable or organ chip is installed on the device. So same device, different consumable is able to run this multi-organ example I'm going to talk to you about. And the reason for talking about this example is that it demonstrates how one can take a pre-existing model and bring it and onboard it into the system. So here we use the Epithelix Mucil Air model and combined it with a liver model. We then scaled the two organs to ensure appropriate communication between the organs, and then undertook co-cultures. And I think scaling is an important aspect. It's important in single organ models, but I think it becomes even more important when we move to these multi-organ systems. And we've started to see many groups around the world publishing on different scaling methodologies. Here you can see a publication from the Sirit group at MIT looking at scaling and modeling of multi-organ platforms. And I think this is a very important area that needs particularly to be considered when we're building these multi-organ systems. Inappropriate scaling is likely to lead to results that are not readily translatable and not readily understood. So here's some of the data from the lung liver model that we created. So you can see in the upper left quadrant a schematic of the organ on chip used, and here we had the lung model housed in a transwell and then the liver housed in a compartment below with media flowing between the two. And in this case, it's particularly important to have the media flow, as in this configuration, the primary human hepatocytes would not survive and remain functional without the flow. So the lung and the liver could be cultured together once optimized cell culture medium conditions had been found, and indeed they could be cultured for many days. And this model was subsequently used to test a number of articles or compounds of interest to the partner in this study. So the last activity I'd like to tell you a little bit about was our human body on a chip model, and that was recently published in scientific reports. So here the goal was to run up to 10 organs, have them interconnected continuously, and have those cultures survive for 1 month. And in doing this exercise, we took that in a modular fashion. So we started with lower order systems, 4 organs and 7 organs, before moving to the larger 10 organ system. You can see in this slide on the left-hand side, you can see an image of the platform, and what you will notice from the platform, again, hopefully, is that it incorporates organ models and tissue models in a variety of formats. So you can see there are some transwell formats, and those can be of different sizes, and also there are some proprietary formats. So, for example, our liver on a chip system was used here. So the platforms built here allow a mixing and matching of different organ chip formats. In the center, you can see the scaling rationale that was used here. So in these platforms, the flow to each organ was scaled relative to cardiac output, and you can see the percentages there given in the figure. And on the right-hand side, you can see data for the 7-organ model, which was composed of liver, gut, lung, endometrium, heart, pancreas, and brain. And here we're looking at a single functional readout for each organ, and that particular experiment lasted for 3 weeks. And to finally give you an example of some of the other models that have been built, indeed by us and by collaborators, a model is currently being developed at MIT looking at gut and gut microbiome and how that relates to the liver and also relates to the brain in the model of Parkinson's disease. Collaborators at the University of Pittsburgh developed a liver metastasis and dormancy model looking at the hepatic niche. And lastly, in our research group, we built a hepatitis B full viral life cycle model using the chip, and have subsequently gone on to use that in some of our own discovery programs. And the last point that was mentioned in the introduction, but I think is worth mentioning again, is that CN Bio is involved in a collaboration with the FDA using our liver on a chip devices. And I think that this is very beneficial for both organ-on-chip vendors and also for the FDA, it allows us to engage in a truly collaborative piece of work to really help to understand where these systems might be of value for regulatory sites.
Wonderful, David, thank you for that. So again, a quick tour through, but I think what we've really tried to do for you today is to share some of the publications which we hope will serve as a resource and maybe an inspiration for some of the research and experiments that you're seeking to do in your own labs. I know that David and members of our team who are co-authors on those papers would be more than willing to answer questions or to dive into the details of how those types of experiments and protocols could be operated in your own research. So just before we finish, we do just have one final polling question. And for this again, Ed is the man with the technology.
Thank you, Dr. Skeets, and that is correct. We do have one final poll question for our audience today. I have launched that, and again, viewers can vote on that in real time. Here, we would like to ask, what is the main barrier to using organs on chips for more frequently in your research? And the available options for this poll are potential for non-specific drug binding, throughput, practicalities of adoption, for instance, lab requirements, expertise, training, etc., lack of suitable biological models or assays, or is it cost being the main barrier to using organs on chips more frequently in your research? We do appreciate everybody voting on this poll. Again, one last final friendly reminder that you can vote on this poll right now live in real time, so we do encourage everyone to do so. Again, what is the main barrier to using organs on chips more frequently in your research? And the options here being potential for non-specific drug binding, throughput, practicalities of adoption, lab requirements, expertise, training, etc., lack of suitable biological models or assays, or is it cost? And it looks like most of our audience members have voted, so I'd now like to share those results with everyone. We have 26% of the audience voting cost as their main barrier here, another 26% voting lack of suitable biological models or assays, 23% of the audience voting throughput, 19% voting practicalities of adoption, and 6% voting potential for non-specific drug binding. Again, thanks to all of you who voted. We have been receiving quite a few questions, and so why don't we go ahead and jump into that. I'd like to thank both our speakers very much for that insightful presentation, and I would also like to invite our audience to continue sending in those questions or comments using the questions window for the Q&A portion of today's webinar. The first question here asks, is it possible to use diseased tissues in your devices, or do you always work with healthy cells and tissues?
So thanks, Ed. I'll answer that one. So it is possible to work with both healthy and diseased tissues in the devices. One thing to note, and this is a particular example from the liver, is that sometimes diseased tissues can be difficult to culture. So, for example, it can be quite challenging to isolate diseased hepatocytes. So one strategy that we've taken and that you saw during my presentation is that we will often take healthy cells, place them in the chip, and then produce a diseased phenotype by adding a disease-causing agent. So maybe a virus or fat, for example. So there are different strategies. I think another point that might be worth making here is that our devices and certainly others can also be used with stem cell derived variants. So, for example, stem cell derived hepatocytes. So that might be another way for people to generate a diseased phenotype by taking an edited stem cell and forward differentiating it to a hepatocyte or other lineage, and then seeding those into the system.
Excellent. Thank you very much for that answer, Dr. Hughes. Another question here. Do the Physiomimics chips differ from the prior liver chips? For example, for the liver, have the media volumes, well dimensions, or consumables been modified?
Biologically, we believe that the Physiomimics chips are similar to the older generation of liver chips, but very importantly, what has changed is that the old liver chip platforms used to have some reusable and some consumable components, but with the Physiomimics platform, we've moved to a completely disposable chip or cartridge. So we believe that we get biological similarity, but with significant usability improvements.
Excellent. Great. Thank you for that answer as well. We do have another question here from another audience member asking, so the plates of CN Bio are not composed of PDMS components, which is a benefit regarding pharmacokinetics, as PDMS absorbs lipophilic small molecules. However, is the material that CN Bio uses for their plates completely inert to absorbing small lipophilic or hydrophilic molecules? And also, what about other molecules such as biomarkers, proteins, or medium supplements?
So the material used for the plates is highly resistant to absorption of small molecules. Obviously, in any system where there is surfaces, there is some potential for protein binding, but again, we would consider the protein binding to be no worse than would be found in a regular well plate. You know, I think another point to make is that it would be wrong to say that any system is completely resistant to non-specific binding. Even well plates will show some binding tendencies with drugs that are very, very lipophilic. So it's an important point, and we would consider that our systems are very inert, but I would say that no one can say their system is completely inert.
Okay, excellent. Thank you. Thank you. Another question here, are the plates microscopy ready, more specifically for live cell imaging? In the systems that I showed, imaging is achieved by removing the scaffold from the system and then taking that to the microscope. So live cell imaging is not one of the primary endpoints that we would tend to use in the system, but imaging is certainly possible. Great, fantastic. Thank you for that answer, Dr. Hughes. Another question here. It seems that endocrine organs are important to include to recapitulate in vivo response. I noticed that you had a pancreas model included in your 7-organ example. Is this actually function to secrete insulin and glucagon?
So in the 7-organ example, the pancreatic model, which I think was composed of primary islets, it did indeed secrete insulin. That was the parameter which was measured. But in that experiment, we did not solely use the pancreas as the source of insulin from memory. So yes, I entirely agree with the contention of the question that it is important to include these secretory organs in multi-organ systems. And I guess my comment would also be that I think that there certainly needs to be some further work in this area. Their incorporation is complex, perhaps particularly, say, per the scaling questions that I pointed to earlier in the presentation. So I think this is a valuable and important area of research, and I still think there's some research to be done in this area. probably worth saying at this point that the information that is presented on the 7 and 10 organ platforms, I think should be viewed as more of a scientific proof of concept, and that it was driven by the way the funders wished the program to be performed, and certainly there would be a lot more analysis one could do with those kinds of platforms, and so I think the paper should be viewed as the starting point, as it were.
All right, excellent. Thank you for that in-depth answer there, Dr. Hughes. Another question for you here. Is there a way to create an in vivo-like organization with LSECs forming a barrier in front of hepatocytes, or do they simply mix up?
That's a really excellent question. That's something that we have thought about much in our research. It's clear that to really accurately mimic the histoarchitecture of the liver sinusoid, one would require a fenestrated layer of cells in front of the hepatocytes. If we look in the literature pertaining to hepatic culture and multicellular hepatic culture, we can see different systems actually that seek to do this. We can see, for example, there would be spheroid models where the Llse might be mixed together with the other cells, going all the way across to much more, say, structured two-layer models where you would perhaps array the Llse on one side of a membrane and the hepatocytes and, say, other cells on the reverse side of the membrane. In the CM Bio device, depending on how one might set the experiment up, I think one could achieve either the mixed phenotype or some degree of layering of Llse into the channel. And I think an important point to consider here would, when deciding which of those models might be appropriate for the research or application or assay that was interested in, is there's clearly a trade-off between the complexity of the model and the ease of using the model versus how faithfully it would recapitulate the histoarchitecture. And I think it's important to consider, whether you really need that very faithful recapitulation, because, say, you may pay a price for that in terms of not being able, having a slightly lower throughput and things like that. So I think the Llse question is a good one to illustrate how, when you're building these kinds of assays, you might want to think about, complexity versus, say throughput and what, you really want to determine with the assay.
Absolutely, and thank you very much for that, viewer, for that answer, and thank you again, Dr. Hughes, for going into great detail with that. It's much appreciated. Another question from another audience member here for you, asking, in the multi-organ models, I assume the optimization of the medium is a challenge, as different cells or tissues require different media compositions in traditional culture systems. How far away are we removed from using a complete human blood plasma matrix, and would this be possible?
So that's an excellent question, and being part of the DARPA project provided a really interesting window into how to approach this problem of optimizing a medium for multi-organ cultures. The way that we approached the problem was to be systematic and to take the media compositions that the various organs were cultured in that we could find from literature, and then undertake essentially comparisons and start to look for components which may cause problems between different organs, and then iterate towards a composition that we felt would support all the organs. I think it's very important, and indeed this is what we did. We came up with different compositions, we tested them on single organs, and then we tested them on combined organs. And that gave us a great deal of knowledge and provided a way to triage and remove compositions which had negative effects on single organs before doing multi-organ experiments. The question, one question which frequently arose was the presence of serum in the medium. We would have liked to run a completely serum-free medium, but in the end, some serum was required. A really interesting paper, actually, very recently published by the CIRIC group, looked at optimization of media composition for different organs, and I would recommend people to search out that paper and look, because it's a really good exposition of how to design these medias for multiple components. Lastly, to answer the question on how far away are we from a human blood plasma substitute, I think we're still some way away from that, but I think we do see some interesting trends in this field, such as more people starting to add human serum albumin in cultures, and also to start increasing the concentration or control the concentration of protein to think about creating accurate bound and more relevantly unbound or free drug concentrations in these systems.
Great. Thank you very much again for that answer. Another question here for the panel. How do you choose the most appropriate medium in the multi-organ device? Could you share your optimized protocol?
Yeah. So certainly. So two things to say on that. I think the previous answer talked a little about how to find an optimized medium. The medium composition that was used in the experiments for the 7 and 10 organ systems is published in the scientific reports paper. We have also published some work looking at optimum medium compositions for hepatic co-cultures of hepatocytes and quiver cells, and indeed also, as I say, the recently published paper from the CIRIC group looking at optimization in three organ models, and that was liver, cardiac, and gut, provides an interesting example of the optimal media compositions for those systems. I think one point to make is that clearly there will be a different optimal media composition for a multi-organ system that is composed of cell lines as opposed to one which might contain primary cells. I think our experience has taught us that primary cells, being the more faithful mimic of the in vivo, will require a little more media optimization than perhaps a model composed of carcinoma cell lines will.
Yeah, and I think importantly to say to everybody, wherever possible, we are not keeping this information on media as proprietary information. We're making this available through the published literature wherever we have relevant information to share it, either in the public domain or provided along with protocols where we have those for turnkey assays on our PhysioMimics platform.
Excellent. Thank you both for reiterating that point. I do believe we have time for one final question here. Has the multi-organ on a chip system been used to study tumor metastasis, and do you have any plans to do so?
So that's a great question. Metastasis would clearly be a very, very good example of where two organs and the intercommunication between those organs is very important in recreating the pathology here. So to date, our collaborators at the University of Pittsburgh have studied metastasis only using a liver tissue model. There was fluid flow in that liver tissue model, but there was no second organ. I think there would be some really interesting models one could build, and I think the work from Pittsburgh provides a really excellent starting point for that kind of research.
Absolutely, absolutely. Well, thank you to both our speakers very much for those answers. We've reached the end of the question and answer portion of this webinar. If we weren't able to attend to your questions today, the team at CN Bio may try to follow up with you after the webinar. If you have further questions, please direct them to the email address showing on your screen. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. Also, a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. I just sent everyone a link in their chat box. You can say thanks to our speakers via LinkedIn using that link. So I do encourage everyone to join us once more and thanking our speakers. Thank you again, Dr. Emma Skeets and Dr. David Hughes for a wonderful presentation. We hope you found this conference informative. Have a great day, everyone.